In this lecture, we're going to be talking about the different cell types that we can find in organisms, the organelles that can exist in cells, and then we'll do a brief intro into viruses. All of this is going to be introductory, nothing too complicated. So let's get started. Prokaryotic cells are the earliest known examples of cells. They're the most primitive cell types on the planet, and they exist in abundance to this day because these are bacterial cells. Now, they do have a phospholipid bilayer. This is what is commonly known as the cell membrane. They usually multiply or clone through binary fission. They don't do reproduction like advanced animals and other organisms do. They don't have any organelles. They have their genetic material just clumped together in the center of a cell. It's usually a single chromosome. It's usually circular DNA in nature, meaning that if you just stretched it all out, it would be a gigantic circle of DNA. These cells are incredibly small. If you compare the largest prokaryotic cells to the smallest eukaryotic cells, the eukaryotic cells are still much larger in most cases. We can have multiple methods of movement that are used by prokaryotic cells. For instance, we can have a large whip-like tail called a flagellum. We can have little hairs all around called cilia, and these all move in unison to move the cell around in a liquid environment. On this diagram, we see these little things called fimbriae, and these are actually used for attaching to other cells. And you can have a transfer of genetic material, but it's not like reproduction. It's a transfer of some genetic material. It can't result in the production of a new organism. And there's lots of theories as to why bacteria do this, but it's believed that this actually allows them to kind of evolve and become stronger as a group. Eukaryotic cells, by comparison, are much, much larger. In most cases, even a small eukaryotic cell is 1,000 times larger than a prokaryotic cell. They're also more complex. Eukaryotic cells have many, many organelles, multiple copies of each in a lot of cases. Eukaryotic cells have a phospholipid bilayer or cell membrane just like prokaryotic cells, but eukaryotic cells also have a phospholipid bilayer around each organelle. Sometimes they have two phospholipid bilayers around an organelle. Usually, eukaryotic cells do not move, although there are some examples, some exceptions. For instance, a human sperm cell is capable of movement. Another difference is that eukaryotic cells have linear DNA, and usually many, many chromosomes, not just one. These are just a few of the examples of the differences between prokaryotes and eukaryotes. Now, from an evolutionary theory standpoint, it's believed that eukaryotes may have evolved from prokaryotes. What may have happened is that two prokaryotic cells, one large one small one, were living next to each other, and the larger one engulfed the smaller one, and the first eukaryotic cell happened. They were able to live symbiotically, and they were able to produce more copies of themselves in that way. Either way, eukaryotic cells are very recent from an evolutionary standpoint. It's believed the first eukaryotic cells maybe existed about a billion years ago, whereas the first prokaryotic cells may have existed 1.5 to 2 billion years ago. This is all theoretical. Of course, no one actually was around then to tell us the truth. Now, one of the key features of a eukaryotic cell are the organelles. And in order to remember what each organelle does, it's actually useful to think of the cell as a large corporation with a product that they produce. Or maybe many products, but either way, they're producing a product. And just like a large corporation, there's multiple aspects to how that corporation operates. Now, the headquarters of a corporation is where all the most important information is stored. The schematics or blueprints for every product being made is there. Well, that would be the nucleus in the case of a cell. The nucleus is where the chromosomes are stored, the genetic material, the DNA. This is the master plan of everything that can be made by this company. This is where the DNA is replicated. If we need to make copies of that master plan, then it's going to happen here. This is also the site of transcription. Transcription is the first step in the production of protein. And basically what it is we take our gene, our DNA gene, and we want to make a copy of it. And that copy will be used to make the protein. Consider this. Let's say you are up in Cupertino, California, and you work at the Apple headquarters. And you have access to the iPhone X squared, the next iPhone. You have access to the phone and you have access to its schematics. And it's your job to get a factory somewhere else to start making it. Do you hand them the only existing phone and the only existing schematics? Of course not. You make a copy of the schematics and then they use that. You would never give away the only master copy. If you consider another example, for instance, the formula for Coke, for Coca-Cola. It's rumored that not only is there only one master plan, it's not even it's divided in half and the two halves are stored separately from each other. No one person has access to both halves of the formula for Coca-Cola. It's more difficult than nuclear missiles. Either way, the DNA is stored in the nucleus, and the purpose of the nucleus is to either start the creation of proteins or to make more DNA so that we can make more cells. Now, the nucleus is wrapped in a double bilayer, which means 2 phospholipid bilayers, and there are pores embedded in that double bilayer that allow transport of raw materials into the nucleus and transport of produced materials out in the nucleus. Now, there's technically an organelle inside the nucleus called the nucleolus. The nucleolus produces ribosomes. This is a type of RNA called ribosomal RNA that helps with protein production. And I equate the ribosome to the factory worker. This is the person that's actually in the factory putting the product together. The nucleolus is kind of like the training facility. It's the one that's going to take the factory workers, teach them how to do their job. In this case, it's making the ribosomes, and the ribosomes will go off and do protein production. Now, transcription, this is the copy machine. This is how we're going to take our master schematic and we're going to make a copy of it so that it can be used in the factory. In this case, we're going to make a copy of our gene. The gene is made of DNA, but the copy is going to be made of mRNA, messenger RNA. Now, translation is the process of reading that messenger RNA and turning it into a peptide chain, or a polypeptide as it's called. And that polypeptide will eventually become a protein. The process of making that polypeptide chain is the equivalent to the assembly line. So the ribosome is going to take the mRNA and read it, and using that mRNA copy of the gene, it's going to start building our protein product. So the ribosome is like the factory worker, the one who's actually putting all the pieces together. They read the instructions and put all the pieces together, and the end result is a polypeptide chain. With a little extra work, that polypeptide chain becomes a functional protein. The Golgi apparatus, sorry, I skipped a step here. The ribosome is, for the most part, embedded in the endoplasmic reticulum, which is what we can call the factory. The endoplasmic reticulum, there's actually two types, rough ER and smooth ER, and we don't need to know the details about them later. But the important thing is to know that the DNA is copied into mRNA, that's transcription. The mRNA leaves the nucleus and goes into endoplasmic reticulum, where it is translated into protein. From there, now we're on the right slide, it goes through something called the Golgi apparatus, which is the FedEx of our cell. So the Golgi apparatus is the shipping and receiving center for our corporation. As proteins enter the Golgi apparatus from the endoplasmic reticulum, from the ribosomes, the proteins are processed, they're labeled for packaging and shipping, and this is no joke. They literally have pieces attached to them so that the cell knows where to take them. The proteins are then shipped off to various locations in the cell and out of the cell, depending on the label that was put on them. And this sounds very complex, and it actually is extraordinarily complex. The amazing thing is that scientists have decoded some of these shipping labels. So scientists can actually take a protein and put a shipping label on it artificially and see what happens to the cell. It's not a whole lot of work been done in this area because it's very complex, but still, it happens. Now, when we're looking at this diagram, I stole this diagram from Wikipedia, and the reason why I've got it up here is it's very helpful. It's a good opportunity for practice going through the steps. So we've got item number 1, which is our nucleolus, number 2, which is our nucleus, number 3 is pointing to a ribosome, number 5 is the rough endoplasmic reticulum, number 8 is the Golgi apparatus. So that's our shipping and receiving center. So that's where we are right now. We've gone from 1 to 2 to 3 to 5 to 8. From there, the proteins can go pretty much anywhere. Now, in order to get through the cell and out of the cell, or wherever they're going, most of the time the proteins are going to be transported on the cytoskeleton of the cell. The cytoskeleton is a network of protein fibers that form the internal structural supports of the cell. They give the cell its shape. There are specific proteins that can actually walk on those fibers. And those proteins transport stuff around inside of the cell. And there's different types of fibers, and different proteins walk in different directions on those fibers. So it's like a bunch of one-way streets with proteins that can walk in one direction or the other. Now, the cytoskeleton would be the equivalent of a highway and rail system for our corporation. It's transporting everything around. We need power for our factory, and in the case of the cell, we have the mitochondria. This would be the power plant. Carbs, fats, and proteins are processed outside of the mitochondria and transported in to be used as fuel. The product is just a different type of fuel, something called ATP, adenosine triphosphate. Now, this is a chemical fuel that the body can use to drive chemical reactions. The interesting thing about mitochondria is that it has its own DNA. Our nucleus does not contain the genes that code for mitochondria. Mitochondria contain the genes that code for mitochondria. And even more interesting is that those genes have been virtually unchanged since the beginning of humanity, and they're inherited maternally, which means that if we look at the mitochondrial DNA of women and we study it globally, we can actually trace back to the very first mitochondrial DNA. And lo and behold, it's in Africa, specifically Ethiopia, which is the cradle of all humanity. And this has been shown in lots of different studies. Now, another interesting theory is how this mitochondria with circular DNA, and remember circular DNA is found in bacteria, prokaryotic cells, how did this mitochondria end up with circular DNA inside of a eukaryotic cell? And much like we talked about the way that the first eukaryotic cells were made, where one prokaryote engulfed another, it's believed that this happened here. A eukaryotic cell, or perhaps a prokaryotic cell, engulfed another prokaryotic cell that was capable of doing all the reactions that the mitochondria can do. And since the mitochondria produces ATP and it can produce more of itself, it was able to help the cell and to replicate itself and become a part of new cells as they were created. Now, the mitochondria, similar to the nucleus, has a double phospholipid bilayer. It really has just one purpose, though, ATP production. That is the primary purpose of the mitochondria. We have another organelle called lysosomes, and these are like the garbage dump. They do the waste disposal and recycling. Lysosomes have a phospholipid bilayer, a cell membrane. They contain lots of different digestive enzymes called lysozymes. They break down everything for either recycling or removal from the body. We recycle a lot of what we consume. Now, it's kind of a gross topic, but it comes up from time to time. Most people think that fecal matter, or poop, is all waste product, the stuff that our body couldn't use. In reality, fecal matter is mostly the stuff that our body just didn't digest. The waste product that our body couldn't use comes out in our urine. Everything else we use. The human body is an incredibly efficient recycling machine. Some people put the estimate to somewhere around 99.9% efficiency. And what that means is that everything gets recycled and reused. Bacterial cells, if they get broken down, even though it's bacteria and bad for us, once we break it down, it's no longer bad for us. Our body takes that bacterial DNA and breaks it apart into the DNA parts that we can use and incorporates it into our own DNA. Why waste energy on making molecules when they've been given to us by other organisms? This is how our body works with all processes. We ingest food, we digest food, and then we break apart the little components so that we can build other stuff out of it. We do the same thing with viruses. We do the same things with bacteria. Anything that our body makes that it doesn't need anymore, let's say a cell is old and it's not working right, our body breaks it down and reuses all the pieces. It's very, very efficient. Now, the lysosomes have a lot of different proteins embedded in the membrane, and they transport lots of different particles into and out of the lysosome. So the lysosome is actually a very complex organelle in terms of the number of functions that it has. The general function is to break stuff down and recycle it or get rid of it. But when you look at how many different types of things it has to break down, pretty much everything that our body isn't using, it ends up being a pretty complex organelle. Outside of the cell, still attached to the phospholipid bilayer, is something called the extracellular matrix. And I like to equate this to the internet or boats that sail across the oceans or planes flying through the sky. This is a network of proteins, carbohydrates, and fats that allow the cell to communicate with other cells. It allows the cells to interact with the environment around it, to understand what's going on outside so that it can react on the inside. So the reason why I say it's like the internet and boats and planes is because the extracellular matrix allows for import and export of materials into and out of the cell. It allows for external communications to go into the cell and communications from the cell to go out. It also serves as a protective layer to the cell. It's harder for bacteria and viruses to actually infect the cell if they can't actually ever touch it. it actually has a lot of different specific functions, and those functions can vary from one cell type to the other. Now, viruses are interesting. Viruses have no lipid bilayer, no mitochondria. They don't use energy or produce energy. They don't reproduce. They don't clone. They're made entirely of protein and nucleic acids, in some cases DNA, in some cases RNA, in some cases both. They can be specific to bacteria or human or monkeys or cats, etc. So some types of viruses can't infect any living thing. They can only infect certain living things. The most important aspect of viruses is how they can make more of themselves. They require a host cell. So viruses infect a host cell, and the host cell actually makes more copies of the virus. So there's an interesting debate that rages on when you get a large group of nerds in a single room. That debate is, are viruses alive? Are they living things? And I think most people, myself included, I refer to them as a living thing, even though technically I don't really think of them as a living thing. It's just easier to think about them as living, even though they don't have any of the properties of a living thing. They don't grow, they don't develop, they don't reproduce or clone, they don't have a metabolism of any kind, they can't make more of themselves, they can't evolve on their own. These are all, the rocks have those exact same descriptive properties, and rocks certainly wouldn't be a living thing. However, viruses, once they come in contact with another cell, can make copies of themselves, can evolve, can develop. So there's a catch here in that viruses require a host cell of some kind. Looking at the life cycle of viruses, we see here we've got a bacterial cell and a type of virus called a bacteriophage. And these are just really cool looking viruses. The picture is literal here. They look like little alien spacecraft. They land on the outside of the bacterial cell, and then a hypodermic needle, an injection right into the cell, shoots the DNA of the virus into the bacterial cell. The virus DNA gets incorporated into the host DNA, and then the host starts making all the proteins that become new virus cells. Those viruses start to get assembled by the host cell proteins, and then eventually the cell fills up to the point that it breaks, and the viruses are released from the broken cell, and they go out and infect new cells. So it's a pretty intense process, and part of the reason why viruses are so hard to fight is because this is the way they operate. They actually require a host cell. For instance, us, we're dealing with COVID-19 right now. Why don't we have a vaccine yet? Well, we haven't found the right set of chemical compounds, the right set of circumstances that are going to prevent the virus from going through this process without killing our own cells. We can kill the virus, sure. We can take Donald Trump's advice and ingest bleach. That'll certainly kill the virus. Unfortunately, it'll kill us too. It's difficult to find the right circumstances to kill a virus, especially since they mutate very, very rapidly. Bacteria are actually easier to fight because they're more similar to our own cells, so we know a lot more about how they operate. Another thing that makes viruses difficult is how small they are. These are made of proteins. You can fit millions, maybe billions of copies of a virus inside a single eukaryotic cell. So their size makes them very, very difficult to study. Even our best imagery equipment, we have problems studying and seeing viral molecules. So anyways, viruses create a lot of problems for us, and we're definitely going to spend some time talking about them in detail later on. Today, this lecture was just to give a quick introduction to the different cell types we'll see, and then of course viruses, which are not cells. If you have any questions, shoot me an email, and we'll see each other on the next lecture.